The coding sequence of most eukaryotic genes is interrupted by intervening sequences, or introns. Consequently, the original version of a eukaryotic gene is very large, difficult to manipulate and virtually impossible to express in any other type of organism. Since mRNA has had the introns removed naturally, it may be used as the source of an uninterrupted coding sequence that is much more convenient for engineering and expression. This involves converting the RNA back into a DNA copy, known as complementary DNA or cDNA, by reverse transcriptase. Thus, when cloning eukaryotic genes, the cDNA version is often used as this lacks the introns. Once the cDNA has been made, PCR can be used to amplify the cDNA and generate multiple copies. This combined procedure is referred to as reverse transcriptase PCR or RT-PCR, and allows genes to be amplified and cloned as intron-free DNA copies starting from mRNA. RT-PCR has other uses. A specific mRNA molecule is only made when the gene for that protein is turned on and expressed. Therefore, extraction and purification of the mRNA gives several mRNA copies of every gene that is being expressed under the particular growth conditions. RT-PCR can then be performed on the mixture of mRNA using PCR primers that match some particular gene of interest. If this gene was expressed under the specific growth conditions, a PCR product will be produced, whereas, if the gene was switched off, none of this particular mRNA will be present and no band will be generated. Carrying out RT-PCR on an organism under different growth conditions reveals when the gene under scrutiny was switched on. This allows analysis of which environmental factors bring about expression of any chosen gene.